Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss some of the important genes involved in embryology. So let's jump right in. Still in the insects, we're leaving the crickets and grasshoppers of order Orthoptera for the horseflies, crane flies, hoverflies, mosquitoes, and other flies of Diptera. As we saw last time, while having two pairs of wings is ancestral for insects, flies reduced their second pair to wings no longer capable of providing lift, called halteres, but which are extremely important for flying. The halteres act as gyroscopes, balancing and guiding the fly in flight. In fact, flies that lack halteres are unable to fly. Other insects have modified their wings. Cockroaches, earwigs, crickets, and grasshoppers, praying mantises, and stick insects have leathery front wings called tegmina, and beetles have hardened forewings called elytra. Fleas and lice have lost their wings entirely. So why does the fruit fly get the tail? Because fruit flies have been invaluable in understanding developmental genes as well as genetics broadly. Let's start this tale of embryology not with our own bodies, but with our mothers. The first few cell divisions of an embryo are controlled by maternal DNA. I am not referring to the portion of the offspring's genome that was inherited from the mother. Instead, it refers to the mother's own genome. Specifically, crucial factors that initiate developmental processes are produced by the mother. Often, these factors are messenger RNAs and proteins. These are very important in early development. In fact, they are pretty much the only genetic factors involved in the first stages of development. During this time, the genome of the offspring is silenced. Meanwhile, the cells, or in the case of the fruit fly, the nuclei of the embryo, will undergo several rounds of division. It's only after a certain point when the maternal factors are degraded and the genes of the offspring are activated, which is called the maternal-to-zygotic transition. This isn't unique to fruit flies. We also observe this in many other model organisms used for research, such as nematodes, sea urchins, zebrafish, and mice. Let's take a closer look at a famous example of the maternal effect. In our fruit flies, maternal cells called nurse cells produce mRNAs for the protein Bicoid, and these mRNA are transported into the oocyte or egg cell. After fertilization, the maternally supplied mRNA is transcribed into the Bicoid protein within the embryo. Since the mRNAs were supplied from one side of the egg cell, the mRNAs are not equally distributed. This means the Bicoid proteins will form a gradient across the developing embryo, with one side having a higher concentration of Bicoid relative to the opposite end. A chemical gradient may seem fairly simple in the context of biology, but this is crucial since this patterns the anterior-posterior axis of the offspring. The bicoid protein is known as a morphogen because its distribution governs the pattern of tissue development by means of activating and repressing other genes, which in turn establishes the identity of these distinct regions. A high concentration of bicoid on one side establishes the anterior end. In other words, the region with the highest bicoid concentration becomes the head. As one gets farther from the site of highest concentration, bicoid concentration exponentially decreases. The region with the lowest concentration will become the tail end of the fruit fly. We know this function of bicoid from observing mutants. If the bicoid gene of the mother is mutated, the embryo will develop tails on both ends. There is no head. Observing such mutants was one of many experiments conducted by Christiane Nusslein-Volhard and her research team. Together with Eric Weishaus and Edward B. Lewis, Nusslein-Volhard received the Nobel Prize in Physiology or Medicine in 1995, making her the sixth woman to become a Nobel laureate. Bicoid is an excellent case of a French flag model of development described by Louis Wolpert. You may wonder how a simple gradient can determine distinct regions across a body, like a head. Well, Bicoid is a transcription factor, and different genes respond to this transcription factor at specific concentrations. Some may be activated or suppressed at a low concentration, while others will only respond at a high concentration. Wolpert described this as a French flag in a case when three genes are activated at progressively decreasing concentrations. Such a pattern is initiated by a morphogen. In this case, a gradient provides positional information. The distance between the cells and the source of the morphogen determines what genes they will activate. However, some patterns can emerge spontaneously. These don't need a morphogenic gradient, they are self-organized. A good example of this in ontogeny is the reaction-diffusion system. This process is responsible for stripes, spots, and spirals in zebras, cheetahs, and giant pufferfish, respectively. In one fascinating example, a team of researchers hybridized the white-spotted char, Salvelinus leucomanus, with the mesu salmon, Oncorhynchus mesu. The former has light spots on a dark background, while the latter has dark spots on a light background. However, hybrids of the two have neither pattern. They have a bizarre dark labyrinthine pattern on a light background. Why? The answer involves English mathematician and computer scientist Alan Turing. He authored a famous 1952 paper, The Chemical Basis of Morphogenesis, that details the mechanism of stripe or spot pattern formation from an initially homogeneous state. This paper describes two molecules we'll call A, the activator, and B, the inhibitor. A is produced first and activates the production of both A and B molecules. However, B molecules inhibit the production of A molecules. So first, A molecules are produced and diffuse homogeneously throughout the tissue, and the production of those A molecules also produces B molecules. In this example, the B molecules are better at diffusing through tissue than A molecules are. Thus, pools of A molecules will quickly become surrounded by B molecules, preventing the spread of A molecules. This results in a spotted pattern. What if we flip the diffusion rate for the molecules? If A molecules spread faster than B molecules, then A molecules won't merely be confined to individual spots. The A molecule spots will leak out, running into other spots and connecting them. This results in a labyrinthine pattern. Or you can imagine a very narrow strip of tissue, like a tail or snakeskin, where there isn't enough room to fully form a pattern. In those cases, there would only be enough room to form a stripe. The relative diffusion and reaction rates are therefore responsible for many of the animal patterns we see. To return to our salmon question, whether the embryo is initially dark with light spots or light with dark spots doesn't matter. Either way, our molecule A spreads much faster than molecule B, such that molecule A initially starts as spots, but comes to eventually surround molecule B. That's why when the two species are hybridized, a labyrinthine pattern results. It's the midway between the two endpoints. What's fascinating about this process is that it's only partially genetic. The components involved are products of genes like proteins. However, the pattern itself cannot be said to be genetic in the same way the amino acid sequence of a protein is said to be determined by the order of nucleotides in DNA. There is no single gene for zebra stripes. There is no genetic blueprint which dictates where each stripe should be. Instead, the relevant genes are involved in timing cell migration. The dark stripes of zebras result from melanocytes that migrate from the neural crest. The common zebra has 26 stripes, the mountain zebra has 43, and Grevy's zebra has 80. British evolutionary biologist Jonathan Bard figured out that the striping pattern results from the stripes initiating about every 0.4 millimeters along the embryo, but at different times. If the stripes initiate early in ontogeny, then the adult zebra has fewer but larger stripes because the embryo is smaller. If the stripes initiate later in ontogeny, then the adult zebra will have more but thinner stripes because the embryo is larger. Indeed, this is exactly what happens. Stripes initiate on the common zebra at 21 days, the mountain zebra 28 days, and Grevy's zebra 35 days. Turing mechanisms are not just about the spots and stripes on the skin of animals, it goes much deeper than that. In vertebrate development, embryonic cilia sweep in a clockwise manner, causing the morphogen nodal to be swept leftward, turning on a different set of genes on the left side of the body compared to the right. This isn't the only example of a morphological pattern established by a Turing mechanism. Feathers, hair follicles, the branches of your lungs, the ridges along the roof of your mouth, and even the patterns of your fingers were first established as spots and stripes produced by Turing mechanisms. Another well-known morphogen is retinoic acid, a form of vitamin A, which is involved in the formation of somites, limb bud initiation, and the formation of heart atria. And there's sonic hedgehog that's involved in patterning the central nervous system, limbs, digits, and many other parts of the body. In general, for large-scale patterns like the dorsoventral or antero-posterior axis, these have to be laid down early in ontogeny because morphogens cannot diffuse very far through tissue. More specific and complex tissue differentiation can only occur later once the embryo has grown larger. Whereas the offspring initially starts as a unicellular zygote, it quickly divides into a compact multicelled morula, then a holoblastula, and then a gastrula. Once gastrulation occurs, in which ectoderm, mesoderm, and endoderm are differentiated, Hox genes start getting activated. Understand that homeobox genes encode homeodomains, which are proteins that bind to regions of DNA and affect their transcription, and Hox genes are one set of homeobox genes. Remember from the lamprey's tale that all jawed vertebrates, or gnathostomes, have 4 clusters of Hox genes. Bilaterally symmetric animals originally had only one Hox cluster, as protostomes, echinoderms, and acornworms, lanceolets, and tunicates still have. However, early vertebrates duplicated their Hox cluster, so the ancestors of hagfish and lampreys had 2 Hox clusters. Later, 2 different species of stem gnathostomes hybridized, resulting in allopolyploidy, and this left us with 4 total Hox clusters. The clusters are named Hox A, Hox B, Hox C, and Hox D. The genes in each cluster are arranged linearly from 1 to 13. No cluster has all 13 because having duplicates of a gene means one or more can be lost without consequence. As it happens, your DNA exists in an initially highly compact state where the strand is wrapped tightly around proteins called histones. Retinoic acid, that morphogen we met earlier, causes the gradual unwinding of DNA, allowing the strand to be accessed by RNA polymerase and transcribed into mRNA. Tampering with Hox gene expression changes the identity of relevant sections. For example, the first vertebra of a mouse is the atlas, and the second is the axis. These vertebrae have specific shapes allowing for head movement, but the third, fourth, and fifth vertebrae all have similar generic shapes. Interestingly, when the HOXA4 gene is deleted, the third vertebrae develops like a second axis vertebra. It's as though the vertebra was tricked into thinking it was developing closer to the head than it actually was. A mutation where the identity of a segment is changed is known as a homeotic mutation, and this idea was originally formulated by English biologist William Bateson in his 1894 book, Materials for the Study of Variation. One particular type of homeotic mutation involved a bee being born with legs where its antenna should be. Later, this mutation would be attributed to a Hox gene called antennapedia, which is homologous to mouse genes HOXA6, HOXB6, and HOXC6. The first homeotic mutation studied in the lab was the identity replacement of fruit fly halteres with fully fledged wings, which was caused by mutations in the Hox gene ultrabithorax. American biologist Thomas Hunt Morgan pioneered the research, and from his lab also came the discovery that genes are carried on chromosomes, sex-linked traits, genetic linkages and linkage maps, and chromosomal crossover, among other things. Most of what we know about genetics started with work on fruit flies. It's easy to imagine someone in 1910 complaining that tax dollars are being wasted on fruit flies, in much the same way that Ohio Representative Jim Jordan whines about money spent on plankton research today. But the medical genetics we take for granted today began as embryologists peering down a microscope attempting to determine whether a fruit fly's eyes were red or white. you can never know in advance where the applications of some research are going to carry you. For fruit flies, they only have 8 Hox genes, and these Hox genes are separated into 2 clusters. The first cluster has 5 genes, and the second cluster has 3. The fruit fly larva initially has 17 segments. These are decreased as the fly molts, but that's not relevant now. And Hox genes control the fates of those segments by generating morphogen gradients like the ones mentioned earlier. So when something goes wrong with a Hox gene, the body segment in question thinks it's the wrong segment. That's what happened with ultrabithorax. Normally, the segment in which it's expressed knows it's the third thoracic segment, but when a mutation knocks this function out, the relevant segment is misled into thinking that it's actually the second thoracic segment, thereby generating a second fully fledged pair of wings. So, Hox genes pattern the bodies of animals, and homeobox genes, more broadly, are distributed all across eukaryotes, from animals to fungi to plants. Now let's look at homeobox genes in an evolutionary context. Fascinatingly, the order of Hox genes in fruit flies matches exactly the order of homologous genes in mice and humans. In the nearly 600 million years of separation between us and fruit flies, the order of Hox genes has remained the same. And as expected under common ancestry, each Hox gene is more closely related to its homologue in other species than to other Hox genes. For example, our Hox1 genes are more closely related to a fruit fly's Hox1 gene than is to the human Hox2.
Spit on that thing!
Furthermore, some homeobox genes have retained their function all that time too. The homeobox gene PAX6 regulates eye development across bilaterians, from humans and mice to fruit flies to snails. Another homeobox gene called Tin Man regulates heart development in fruit flies, and, of course, vertebrates have a homologous gene. The human version is less humorously called NKX2-5. But while there is a large degree of overlap in spatio-temporal expression for Hox genes across distantly related animal phyla, that shouldn't lead us into the trap of thinking all homeobox genes do the same or similar jobs in different clades. There is actually quite a large degree of variability in what functions a homeobox gene might have. Let's look first to our side of the bilaterian divide. The linear arrangement of Hox genes is more or less the same for vertebrates, tunicates, lancelets, and acorn worms. But what about echinoderms? Remember from our discussion of echinoderms in the ragworm's tail that they exhibit bilateral symmetry as juveniles and transition to pentameral symmetry in adulthood. You might wonder then, does the echinoderm larva exhibit linearity in Hox gene expression from anterior to posterior like us, or fruit flies do? Well, sort of. Remember from the aforementioned video that echinoderm larvae have originally 5 coeloms that get radically reorganized through ontogeny. One of those coeloms is the curled gut. The anus is right below the mouth, so the Hox genes are largely expressed linearly along the gut. There isn't really an antero-posterior axis in the larva because of this curling. So what's going on with adult echinoderms? We must consider then 2 broad hypotheses for adult echinoderms. Either the ancestral deuterostome arrangement has been completely dismantled and reassembled into a novel body plan, or there is still some underlying deuterostome body plan that's just undergone some adjustment. A few different hypotheses have surfaced to explain this. First is bifurcation. Imagine that our more traditionally wormy deuterostome was split at the posterior end into 2 legs and also sprouted 2 arms. Second is circularization. The homeobox genes start at 1 arm and wrap around the body. Third is duplication. Each arm is considered a separate axis, where the homeobox genes start at the center and move outward towards the ends of each arm. Fourth is stacking. The homeobox genes are arranged from either the oral to the aboral end or vice versa. The 2023 paper, Molecular Evidence of Antero-Posterior Patterning in Adult Echinoderms, rejects all of these hypotheses to some extent. The stacking hypothesis is closest but still incorrect. Rather than the homeobox genes starting on the oral end and then going upwards, they actually start at the ambulacral grooves. The authors term this the ambulacral anterior model. As they write, quote, "In this model, the midline of each ambulacrum expresses the most anterior bilaterian molecular identity equivalent to the forebrain and proboscis in vertebrates and hemichordates, respectively. The midlateral regions on either side of the nerve cords, including the ectoderm wrapping the podia, share patterning similarities with more caudal ectodermal territories of hemichordates and chordates, down to the collar and midbrain, respectively. Finally, the boundary at the edge of the ambulacral ectoderm displays the most posterior molecular profile corresponding to the collar trunk boundary of hemichordates and the midbrain hindbrain boundary of vertebrates." Close quote. The authors propose that this arose by modification of an ancestral deuterostome axial program during echinoderm evolution. As for other phyla, the function of one homeobox gene can be substituted by the function of one or more other genes. Let's return to Bicoid, for instance. Bicoid mRNA is deposited into the egg after fertilization in fruit flies, but in flower beetles and parasitic wasps, mRNA of the genes activated by Bicoid is deposited into the eggs prior to fertilization. So there's no Bicoid in the egg at all. In other words, differential expression of those genes compensated for the action of Bicoid. As biologist Tangui Chuard noted, quote, "It all feels very counterintuitive. Maybe it is because man-made machines are sensitive to individual component failure that biologists tend to assume that genetic networks are similarly constrained and that changing their wiring piecemeal would mess up the whole system." Close quote. But of course, living organisms aren't machines. This oft-used analogy fails to capture dynamic systems. Can we find examples in nature of a transitional phenotype bridging two very different ones? Absolutely. Let's take the sexual differentiation system of yeasts. Most yeasts have two mating types, A cells that express A genes and alpha cells that express alpha genes. In the human pathogen Candida albicans, A genes must be turned on by the DNA-binding protein A2 before they can be expressed. But in Baker's yeast, Saccharomyces cerevisiae, A genes have to be turned off by the repressor protein alpha-2 because their default position is on. How can it be the case that Baker's yeast works oppositely of Candida and doesn't even have the necessary A2 protein? It turns out that there is a third DNA binding protein called MCM1 that can bind to either A2 or alpha-2 depending on the yeast species. In a yeast used in the cheese industry, Cluiveromyces lactis, MCM1 binds to both A2 and alpha-2, bridging the gap. By having this functional redundancy, more derived yeasts could lose ancestral proteins without deleterious effects. Even within a genus, there can be radically different developmental paths. Take the sea urchin Heliosidaris. H. tuberculata begins life as a planktonic pluteus that later metamorphoses, while H. erythrogramma directly develops from an egg to an adult in just a few days. How could this have happened in a mere 4 million years? It turns out that both modes are a response to the question of how to build an adult urchin that sticks to a rock and how to survive youth in open water. Urchins with pluteus larvae produce many small eggs where most don't survive to adulthood, while direct developing urchins produce just a few large eggs that are more likely to survive. Since having a pluteus larva is ancestral for urchins, it could be the case that natural selection favored direct development for some urchin species, but biologists like population geneticist Michael Lynch aren't convinced by this answer. Instead, they attribute the role of chance, i.e. genetic drift, to such changes. Regulatory networks can tolerate point mutations, duplications, and deletions much better than protein-coding genes can. They can cause protein-binding sites to appear or disappear without a loss of fitness. As Lynch has written, quote, "A growing body of evidence supports the idea that many aspects of complexity at the genome, molecular, and cellular levels in multicellular species are likely to owe their origins to these non-adaptive forces, representing little more than passive outcomes of the unique population genetic environments that are presented by such lineages. Genomic features that are consistent with such evolution include the preservation of duplicate genes, the evolution of modular gene architecture, the establishment of introns in untranslated regions, and now the evolution of network architecture and redundant regulation. Close quote. So that's the fruit fly's tale. Model organisms like fruit flies and mice have greatly elucidated the genes that underlie our development, and there is an enormous body of literature documenting the evolution of these genes across lineages. So, thanks for watching, and we'll see you all next time.